Hello, students. This is Dr. Faizan Mirza. Here I am discussing genetics for the A-level biology course of A2. Now, in genetics, we have about seven different type of crosses in your syllabus, and let's just review them one after the other. So, before I go into the detail of these crosses, let's just try to recollect what actually genes are and what's a dominant trait, what's a recessive trait, what is autosomal. Let's just recall those terms so that whatever we are to discuss, it goes in a straight way. So, what is an autosomal dominant trait? We know trait simply means a feature of an organism. The dominant trait means that now for this particular trait, you will have genes and a single allele will be enough for for that trait to be expressed. So that's called a dominant trait. Autosomal, the term autosomal means that the location of the gene or the gene locus is on the autosomes and not the sex chromosome. So the gene locus is on the autosome and not the sex chromosome. The allele, since it's dominant, so it's not recessive here. The allele expressed in phenotype when a recessive allele is present. So yes, it can express itself there, and only one copy of the dominant allele is enough for it to be expressed in the phenotype. For example, there's a trait being controlled by a gene capital A, or capital A, small a. So in both cases, this will be expressed in the phenotype because at least one dominant allele is present there. So a dominant will express itself when it's in a homozygous condition. It will express itself when it's in a hybrid condition as well. A hybrid condition is one in which we denote it with a capital letter and a small letter. This is heterozygous dominant or a hybrid condition. What's an autosomal recessive trait? Autosomal recessive trait, again, the term autosomal mean that the locus of the gene is on the autosome, not the sex chromosome. Since it's a recessive allele, so the allele is not dominant, and the allele will only express in the phenotype in the absence of the dominant allele. So two copies of those recessive alleles must be present together to be expressed in the phenotype. So if we just take this example, for example, both small letters, small a will be expressed in the phenotype when both the recessive alleles are there. So this is the homozygous dominant and the heterozygous dominant and the homozygous recessive. The condition when you have both the same letters being represented together, it's called purebred condition. So this one, the first one, the capital A is homozygous dominant. This is small a is homozygous recessive. Both are considered to be purebred. This one is purebred dominant. This one is purebred recessive. When you express capital A, small a together, it will be called hybrid. It can be called carrier of the recessive trait. It can be called heterozygous dominant because it's having one copy of, or you can say one allele which is dominant. It is having the recessive allele as well. This organism will express the dominant phenotype, but since it's a carrier of the recessive trait, it will be able to pass it on to the next generation. So the offspring will have this allele, and if the other parent also pass on the same allele, there will be a chance of this allele being expressed. How so? Let's just recall these basic crosses that you already must be very familiar with. If you cross a purebred dominant with a hybrid individual, these are the ratios that we get. You get two chances of the homozygous dominant and two chances of the heterozygous dominant being here. So all of them will be expressing the dominant phenotype because all of them will have at least one dominant allele present. If you cross two hybrids together, in that case, the ratio changes. Now you will have 3:1 ratio of phenotypes, which means three of them will have there is chance of 3 out of 4 that at least one dominant allele will be there in the offspring and it will be expressed. And there's 1 in 4 chance that the recessive allele will be passed on from both the parents and the offspring will express the recessive phenotype. In case of both homozygous recessive or purebred recessive being crossed together, 100% offspring will be having the recessive phenotype. In this case, the dominant phenotype cannot be expressed because both the parents were homozygous recessive in this condition. So this was a very simple you can say an example of how the genes work. Now, what do these letters represent? Where are these letters located? These are located on your homologous pair. For example, this individual will have a homologous pair. The homologous pair will have chromosome, one paternal and one maternal, one that this individual received from the father, the other that this individual received from the mother, or one from the maternal lineage and one from the paternal lineage. Now this is small a, for example, is located here. This is the gene locus of the small a. The other small a is present on the other chromosome, the other part of the homologous pair. So these two letters represent this. For example, if the individual is capital A, where is this capital A located? So again, this is the homologous chromosome of this individual. The homologous pair, one paternal chromosome, one maternal chromosome, and this capital A allele, for example, if it's present here, the other capital A allele is present in the same gene locus but on the other chromosome of the homologous pair. Now, if you say that the individual is capital A small a, in this case, the individual will have the same homologous pair, but now this individual received one dominant allele and one recessive allele from each of the parent. So, for example, if this capital A was present, let's suppose its gene locus is here, for example, or the small a is present on the other homologous chromosome. So, I have just changed this the gene locus here just to tell you that the gene locus is present on the chromosome of for the same gene. The gene locus is the same, so the individual can either be homozygous dominant or homozygous recessive or heterozygous dominant in that case. So this where is the gene located? The genes are located on the chromosome, on the chromatid, and the homologous chromosome pair is having the same gene, but a different allele might be present there. So the crosses that we have in our syllabus, as far as genetics is concerned, there are about seven different crosses. The first one being the monohybrid cross. In monohybrid cross, what do we do? What do we do? We just take the example of individuals having one gene, and that one gene is having two alleles. So it deals with a cross involving one gene with two alleles, and one allele will be dominant, one allele will be recessive. And let's take an example of the gene HBB. What is HBB gene? This HBB gene codes for the beta polypeptide in the hemoglobin. There are two alleles, one is dominant, one is recessive. So HBA is the dominant one, HBS is the recessive one. Now these are the three possible combinations that a person can have. A person could be homozygous dominant for this allele, for this gene, having both dominant alleles. A person can be carrier of the recessive allele. A person can be suffering from sickle cell anemia, where both recessive alleles are present there. So in these individuals, what is the change? The change is in the there's a point mutation. The HBA allele, this is the genetic sequence of the HBA allele. There's a codon CTT that changes to CAT. There's a point mutation on one of the nucleotides, and because of this, a different amino acid get placed into the polypeptide of hemoglobin. So if HBA allele is there, the amino acid that gets placed is glutamic acid. So it is just as abbreviated as GLU. This is the polar amino acid. This allows the hemoglobin beta chain to be folded normally. You can recall how the protein folds because of the polar amino acids, the polar R groups. And if this gets replaced by a non-polar amino acid, which is valine in the same place, so HBS makes a non-polar amino acid to be incorporated to a chain in place of glutamic acid. As a result, the protein gets misfolded, which causes sickle cell anemia. Now let's just review the crosses. For example, if you cross an individual who is a carrier of the sickle cell trait, in this case, I'm taking the female to be the carrier of the sickle cell trait, and I'm taking the male to be the homozygous dominant, which is normal. So what will happen? These are the chances you can make a Punnett square out of it. So there's a chance that both these parents they pass on the same dominant allele. So this individual will be normal. There will be a chance that this individual will be a carrier. Then there's a chance that again this person will be normal, and there's a chance that the next person will be a carrier. So what do we get? We get 50% chance of carrier offspring and 50% chance of a normal offspring. Now let's take a quick example of if this, for example, was a cross between a carrier in female and a carrier male. Both are carriers of the same recessive trait. They both will not be suffering from the disease because they both have at least one dominant allele present. So they are not having the disease, but they can pass it on to their offspring. So when we are having a condition where one gene is having two alleles and the recessive allele is the disease, so what are the chances of the disease being passed on to the offspring? There's 1 in 4 chance. When there's 1 in 4 chance, so 25% chance will be there that the disease will be passed on to the offspring because the parents are the carriers of the same recessive trait. And if we take another example of, let's say, I want to cross two individual who is like one is the individual suffering from sickle cell anemia. This individual will produce gametes both with HBS, and then other individual is a carrier of the recessive trait. So we can again make the crosses, and we can see if this gamete HBA fuses with the HBS gamete, then the individual will be carrier. Then again, the HBA dominant allele fuses with the HBS of the other. Again, you have a dominant feature being expressed. So both of them will be carriers, but the dominant allele is there, but they are both are the carriers of the recessive trait, very similar to the parent. Now, if this HBS gamete fuses with the HbS gamete, that will be the case of sickle cell anemia being passed on. Or lastly, again, the same condition that sickle cell anemia will be passed on. So this is again 1:1 ratio and 50% chance of carrier, 50% chance of the person having the disease. So this is pretty much the same thing that I discussed initially, that if you cross, these are the ratios that we normally get when you have one gene having two alleles, one dominant allele and one recessive allele. And these are the features that we get. So the second type of cross that we get in our syllabus is multiple alleles. What is multiple alleles? In multiple alleles, this involves a cross where genes have more than two alleles, and we have homologous chromosomes. So an individual can have only two of these alleles, one from each parent. For instance, let's take an example of blood grouping. Blood grouping IA, IB, IO. These are the three ways in which we are representing the alleles. IA and IB, the information is given to you. They both are dominant traits. The dominant alleles of the trait, and IO is the recessive one. A person can have any two of these located in the homologous chromosome. Now, similarly, for example, a person B can be IAIA. The person IAIA would have the homologous chromosome like this. This is the homologous chromosome of the individual, and one paternal chromosome, one maternal chromosome. And let's suppose the individual is having an allele I, capital A here, and the other homologous chromosome pair partner from the maternal also is having IA. So this individual is IAIA. In this case, the blood group will be A because the person is having both dominant alleles. Then the person can be IAIO, which means the person will still have blood group A, but because of this O, the person will be carrier of the recessive trait. The person can pass this O to the next generation. Similarly, blood group B can be either IBIB or it can be IBIO. In this case, again, this individual, they both have blood group B, but this individual IBIO can pass the O allele to the next generation. A and B both being dominant. So if they are present together, the blood group is AB. If the person is blood group O, there is no chance other than the person being homozygous recessive for the trait because IO is the recessive allele. Now, if you take an example of these crosses, like what crosses will you, what outcome will you get? For example, if father is homozygous dominant for blood group A and the mother is homozygous dominant for blood group B, so when they are crossed together, all the offspring will be 100% hybrid, having the blood group co-dominance IB together, because A and B will go together and they will give you the offsprings in this case. Now, if you are having any confusion, you can just draw the Punnett square, or you can do the cross yourself and just try to figure it out that this is the ratio that you're getting. If an individual is I, capital A, I is an O, which means carrier of the recessive trait, and the other is the mother, for example, is blood group A and homozygous dominant for it. If you cross them together, and let's see, let's take this is the cross we are getting. One individual will be having blood group A, the other will also having blood group A, 50% chance of carrier, 50% chance of homozygous dominant blood group A. If both parents are carriers of the recessive trait IBIO, IBIO, in that case, 25% individual will have blood group B homozygous dominant, 50% will also have blood group B, but as a carrier of recessive trait, and 25% will have blood group O. Lastly, if an individual is IO blood group O and the mother is blood group AB, if you cross them together, so this is the outcome that we get. So 50% blood group A and 50% blood group B. Let's take a question here. For example, a man of blood group B and a woman of blood group A have three children, and they have blood. One of them is blood group A, the other child is blood group B, the third child is blood group O. What are the genotypes of these five people? So if a father who is having blood group B and a mother who is having blood group A, they can have babies or offspring with blood group A and B, of course. But can they have a individual offspring with blood group O? Yes, very well possible. You will pick the question from here that if a blood group O organism is born, individual is there, that means this person received blood group O from both the parents. That means both the parents, being blood group A and blood group B, must be the carriers of the recessive trait. So they ended up having IO in one gamete and this IO in the other gamete, and the gametes fused together to produce the child with blood group IO. So what are the genotypes? The blood group B will be IBIO, blood group A will be IAIO, and then blood group A and B again will be same as the father and the mother, and blood group O will be recessive IO. So you pick it from the recessive trait and just proceed accordingly. Just in case if you are not familiar with the crosses, that how the crosses are to be done, this is a very simplistic way of drawing the cross. First of all, you mentioned, for example, I'm crossing this purebred dominant A and with purebred dominant B. What would I do? I will just mention the genotype of the parent. These are the genotype of the parent. What is the phenotype? This is blood group A, blood group B. When they form gametes, we separate the homologous chromosome into separate gametes. So we know that half of the gametes will have this dominant allele, half of the gametes will have these. So these are the two type of gametes being produced by the individual who's having dominant trait. Now similarly, the person who is having the other dominant trait, his or her gametes will again be 50%, 50% each. So you will have both gametes of the same IB. Now when you cross them together, you cross the first gamete with the first, then you cross the first with the second, then you cross the second with the first, and the second with the second. In this case, you get all the genotypes here. And once you have genotypes mentioned, you can mention their phenotypes and the blood group which are there, and you can calculate the ratio that how many of them have which blood group. The third type of cross that we have is called sex inheritance. The sex inheritance, we know that sex inheritance, according to the chromosome, is basically dealt not on autosome but on the sex chromosome. For example, humans. Humans are diploid organisms. We have 22 pair of autosomes, which means 44 chromosomes of ours are autosome, and the 45th and the 46th are the sex chromosome. In other words, you can say that males and females are 44 XY being male and 44XX being female. What do you mean by 44XX being female? 44 chromosomes are the autosome. This is the 45th and the 46th chromosome that makes a female of 46 chromosome. Similarly, a human male is 44XY, which means 44 are autosome. The 45th chromosome is X and the 46th is Y. In this case, again, a human diploid male is there. Now, if we talk about the haploid cells, we know that females being 44XX will always produce the same kind of gametes every time. 22 autosomes. We know the gametes do not have any homologous chromosome. They are single chromosome there. So 22 autosome will be there together with one X chromosome. All female gametes will be alike. In human male, although the gametes will be produced in equal quantity, two type of gametes will be produced. Half of the gametes will have 22 autosome with one X chromosome, and half of the gametes will be 22 autosome with one Y chromosome. So when we go to gender determination as far as the chromosome is concerned, so XX chromosome of female and XY chromosome of male, when they go together, so there's a chance of of daughters being born 50% and 50% chances of a son being born or male being born. How is that possible? The females, they receive X chromosome from both the parents, father as well as mother. So both daughters will receive one X chromosome each from their mother and one X chromosome from their father. As far as the males are concerned, they receive X chromosome from the mother and they receive the Y chromosome from their father. A father, under normal circumstances, cannot pass on the X chromosome to their sons. The fathers always just pass on the Y chromosome to the son. That is exactly why the Y chromosome ends up in males in the first place. This brings us to the sex-linked traits. What are sex-linked traits? The sex-linked traits are traits that are having gene locus on the sex chromosome, but the gene is itself not involved in gender determination. And these include the example of genes on the X chromosome. For example, this is the human X chromosome shown to you. The human X chromosome, for example, this gene here enables the kidney to retain phosphate and so on and so forth. You can see several genes here. The last gene is something that we are concerned with as far as our syllabus is concerned. The last gene here on the chromosome X is called F8 gene. The F8 gene controls the production of factor 8 protein. The factor 8 protein is required for blood clotting. The lack of this allele causes hemophilia A. So this brings us to the fourth type of cross that we have. We call it sex linkage. Now in sex linkage, what do we see? We see the cross between two individuals. One of them is having the sex chromosome being a homologous pair and the other is having sex chromosome which is not a homologous pair. So in case of humans, we take the example of F8 gene. F8 gene is called the human factor 8 gene. It is located in the X chromosome. It has got nothing to do with the gender. It is not a gender determining gene, but the trait is passed on according to sex linkage. Now, how is that going on? The F8 gene is having two alleles. One is dominant, one is recessive. Capital H is normal, which is dominant. Small h is hemophilia, which is recessive. Now, a person can have a dominant allele, and one copy of dominant allele is enough to express the normal factor VIII. But if a person is small h, it means both small h must be together for a non-functional factor VIII to be there. But the problem occurs that females and males, they both don't have the same number of sex chromosome. So what's happening here? A female could be either XX, which means homozygous dominant for this dominant allele, and a female could be X capital H, X small h, being a carrier. Why? Because females have both X chromosomes there in their cells. A dominant allele might be present on both X chromosome, or there can be another female having one dominant and one recessive allele being present there on the sex chromosome, or the third could be both small h's there on the H chromosome. Now, since females are having a homologous pair as the sex chromosome, this is why these are the three chances that we have. But this last one, X small h, is a lethal combination. The zygote simply does not develop. What's a lethal gene combination? Lethal gene combination simply means that the fertilization occurred, but the zygote does not develop. And when it does not develop, so we do not have females who suffer from hemophilia A. So a female could either be normal for the dominant allele or could be a carrier for the dominant allele. Now, and the carrier of the recessive trait in that case. And why is that so? Because females have the gene locus of F8 on both the X chromosome, whereas males have the gene locus on the X chromosome, and they do not have the gene locus on the Y chromosome. Y chromosome is a much shorter chromosome. On Y chromosome there is just no gene locus of F8. As a result, a male could either be X capital H Y or X small h y. In this case, the male will be normal, having a dominant allele, or the male could be hemophilic, having hemophilia A because the small h allele got passed on. Now, what about the crosses? This is how the crosses go about. If you cross a normal male here, the normal male being crossed, being X capital H and Y, I took the gametes on the right here, or sorry, on the left here, and the female being X capital H, X small h, I took the gametes on the top. So if you cross them together, we see that the daughters are 50% chances that among the daughters being produced, 50% daughter will be carrier, 50% daughter will be normal. Or in case if you compare with all the offspring, 25% chance being a daughter and a carrier, and 25% chance of being a daughter and being normal. If we look at the Y chromosome being passed on here, so we see that there's a chance of normal son being born and a chance of hemophilic son being born as well. This hemophilic son is being born because the hemophilic son received the hemophilia allele small h from the mother and the Y chromosome from the father. And since this person ended up suffering from hemophilia, it was because of the carrier mother who passed on the small h allele, not because of the father. Now, can this be passed on from a father to the daughters? Yes, it can be passed on from the father to the daughters. For example, if the father is hemophilic, in that case, the father is having X small h on the X chromosome, and Y will not have the gene locus there, whereas the female is X capital H, X capital H is normal female. Now, when you cross them together, there is a chance that all the daughters will have one X chromosome from their father, and the father will pass on the small h allele to their daughters. Whereas the mothers pass on the normal allele dominant to their daughters. As a result, the daughter will not suffer from the disease but will be the carriers of this disease. The hemophilic father, on the other hand, will pass on the Y chromosome to their sons. They cannot pass on the hemophilia disease to the sons because the sons receive the dominant allele on the X chromosome from the mothers, and these sons receive Y chromosome from the hemophilic father. Now, we know that hemophilia cannot be passed on from the father to the son, but through a carrier daughter, this can be passed on through a carrier daughter to the grandson. How is that possible? Because these carrier females are the ones which became these carrier females, and then they pass on the small h allele or the recessive allele to a son through another male who was normal, who was not hemophilic, and the allele got passed on. So this allele that the hemophilic individual received came from the carrier mother. The carrier mother received it from the maternal grandfather. So this individual hemophilic male through carrier daughter ended up passing on the allele to the grandson. So hemophilia can be passed on from a male suffering from the disease to the grandson to the maternal lineage through carrier daughters, but cannot be passed on to the sons. So these are the four major crosses that we discussed. as far as your one gene and alleles are concerned. The remaining crosses deal with when we consider more than one gene at the same time. I'll discuss them in the next video. That's it from my side. Thank you so much.